virus infections of a host are classified into one of two general areas. They can be acute or persistent infections. And today and next time, we are going to be looking at each of these individually. Today, we're going to start with acute infections. So here is a graph, a set of graphs that illustrate the differences between these two different kinds of infections. Again, we're talking about infections of a host with viruses, and what's the outcome? One outcome is an acute infection, which is shown on the top graph. Now, in this graph, we're looking at time on the x-axis, and the y-axis is virus production. So virus production is the blue line. And then the red areas are disease. So you can see the top line is an example of an acute infection where there is rapid rise in virus production accompanied by disease of some sort, and then the infection is resolved, and that's the end of it. That's the definition of acute infection. It comes and goes in a finite amount of time, and examples include rhinoviruses, rotaviruses, influenza viruses. The rest of these graphs illustrate persistent infections of various sorts. This is going to be the topic for next lecture on Wednesday. But the essential element of a persistent infection is that they are not resolved. The virus is never cleared from the host. So you can see, for example, the second line, latent infections. You have periods of virus production followed by periods where you don't detect any virus. And this may or may not be associated with disease. And they're also, so that's called a latent infection because there are periods where we don't see any virus production. In contrast, there are persistent infections where we always see virus produced for long periods of time. For example, the third graph is a persistent asymptomatic infection where we don't get any disease. And then the bottom graph is a persistent pathogenic infection where we have low levels of virus produced throughout infection with disease at the beginning and at the very end. We'll talk about persistent infections next time. Today I want to focus on acute infections. And the best way, I think, to illustrate the principles of acute infection is to go over a few of the better known ones and talk about how they occur and how they are resolved. So that's what we're going to do today. So here are some features of acute infections. There's rapid onset of viral reproduction. There is a short period of disease, and this could be mild or it can be severe. That doesn't matter. The point about being acute is that you have a rather rapid onset, you have production of large numbers of virus particles, and then eventually immune clearance. The immune system kicks in, gets rid of the infection, and that's it. And then hopefully you have lifetime immunity to that virus, and if you're infected next time, you'll know what happens because we've talked about that before. So here's a graph of a typical acute infection. Again, rapid and self-limiting. Those are the hallmarks of an acute infection. Here we have virus growth again on the y-axis. In the x-axis we have time. So virus entry begins at the origin on the lower left. And the red line is virus reproduction, the amount of virus that is produced. And remember, as virus begins to replicate, it encounters innate defenses. And in many infections, these are sufficient to counter the infection. But if the virus titer continues to rise, the innate defenses can't get rid of infection. You trigger the adaptive response. So there's some kind of threshold that's shown here. Threshold level required to activate the adaptive response, which varies according to the virus. Reproduction continues. The adaptive response kicks in. And then eventually, virus peaks, and the adaptive response is able to clear the infection, virus goes away, and there's no more virus left in you. That's another hallmark of an acute infection. And all this is accompanied by memory. All right, so this is what an acute infection is: rapid onset and resolution of the infection. Now, all infections have something associated with them that we have to talk about now, and this is what's called the incubation period. Whether you have an acute outcome or a persistent outcome, in the beginning there's an incubation period, and that we define as that time before the symptoms of disease become clear. Now, that can be a flu-like illness. That's a disease symptom, or it can be a rash, doesn't matter. Whenever the patient or the animal begins to show symptoms or signs of infection, that's the end of the incubation period. During this time, of course, the viral genome is replicating and the host is responding. And of course, if the host is making interferons, then you're going to get flu-like symptoms, systemic organism-wide symptoms that we talked about previously. So that'll be the end of the incubation period. And during this time, very key point here, the virus may or may not be transmitted. Whether transmission occurs during the incubation period depends on the virus. So for some viruses, while the infection is incubating, before you have any symptoms, you're actually shedding virus, and obviously you can infect other people. So that has implications for public health, because if you don't know people are ill, it's very hard to limit infection. Great example, kids in school. They can be shedding virus before they show the rash of measles. So they're going to school, and as soon as they have the rash, of course, their parents keep them home, but by then it's too late. The disease has already spread. In contrast, Ebola virus, there's an incubation period during which time we have never seen anyone transmit infection. And apparently for Ebola virus, you are not infectious during the incubation period. Now, last year and the year before, when we had a big outbreak of Ebola virus in West Africa, many people from other countries were going there to help out. And they would come back home after so many weeks or months of working there. And here in the US, the CDC said, these people are not contagious. If they were potentially infected on their last day in Africa and they come here, they're not contagious because they don't have any symptoms. If they do happen to develop symptoms, then we will hospitalize them and isolate them. But until then, they should be free to walk around. Well, the governors of New York and New Jersey decided they were doctors. Now, as far as I know, neither of them went to medical school, and I'll bet they didn't do very well in science in college. They decided that these individuals were infectious during the incubation period. And so they said, you have to be quarantined. So idiots. There is no transmission during the incubation period of Ebola virus. I don't know why they did that. Many people started to sue as a consequence. You may have heard of a nurse in another state who just went out and said, to hell with my quarantine, I'm not staying home. So this was a big deal. But the incubation period, whether or not you're infectious, depends on the virus. And for Ebola, we've never seen anyone transmit infection to this day. And so all you have to do is monitor people daily who have come back from taking care of people with Ebola infections. And as soon as they show a fever, put them in the hospital. And that's been fine all the time. Now, incubation periods vary in length for different virus infections, as you might expect. They can be as short as a day or two for influenza virus. Rhinovirus is a day or three. Ebola virus has a very long incubation period from 2 to 21 days. And I think this depends on many factors. The host, what kind of immune system the host has, its genetic makeup, how much virus you get infected with, the route of infection. And I think our experience with Ebola isn't that great yet. So we have a real big window of infection periods. But again, during that period for Ebola virus, you are not infectious, although for other viruses you may be. So you see there's a big range, and it gets higher and higher up to a really long time between the time you acquire a papillomavirus and the time you get a wart. So anytime you get a wart on your fingers, hands, feet, elsewhere, wherever you get them, they're usually viral, and the incubation period from the time you acquire them is between 50 and 150 days. Now, as I said, there are many determinants of incubation periods, including your genetics of your immune response and how much virus you get and where. But one of them is roughly some viruses with short incubation periods. In those, wherever the virus comes in, that's when it causes disease. So respiratory viruses like influenza and rhinoviruses, they go into your upper tract and they replicate there, and they start to make disease in the upper tract. So that doesn't take a very long time. In contrast, other viruses, rabies has to go from wherever the dog or animal bites you into your CNS. That takes time. Measles, as we'll see today, has to go from your respiratory tract to the blood to the skin. So maybe that takes longer as well. Now, an important consideration with all infections, whether they be acute or persistent, is that many of them are inapparent. And I think we have talked about this before. I will call them either an inapparent infection or an asymptomatic infection. That's where we have the virus successfully infecting you, but there are no symptoms, there are no disease, yet you can shed virus and spread it. So you appear to be perfectly healthy, but you're shedding virus and spreading it to other people. Now, you may ask, how do we know that this happens? How do we know that there are inapparent infections? And so my answer is, what do you think? How do we know this? How do we know that viruses infect you without causing disease? Certainly by now, you can answer this question, right? How would you know? Yes?
By looking for antibodies.
By looking for antibodies. We do serological surveys of populations, and look, half of the population is seropositive for a virus, but only 10% of the people reported symptoms. We do this routinely for many viruses. We do serological surveys, and we find for many, not all, that the percent seropositivity is much higher than the amount of disease that we see. So that's how we know this. In general, I think this is a property of a well-adapted pathogen. It's something that has been in humans for many, many years. And you may be thinking, well, how many years are you talking about? Well, you know, some viruses have newly entered us, like HIV entered us in the 1920s, Ebola viruses in the 1970s, and so forth. But others have been with us since our ancestors, before we were Homo sapiens, and they passed them down to us. So hundreds of thousands, maybe millions of years. And during that time, they have the chance to adapt and cause less disease. And I think not causing symptoms is a sign of that. A nice example that I like to use is poliovirus, because over 90% of polio infections are inapparent. There's no symptoms. You know, you probably think of, when you hear polio, you'd probably think of paralysis, or maybe you don't think of anything. You should think of paralysis because it's very famous for causing paralytic disease. But in fact, that only happens in 1% of infections. 9% you get some flu-like illness, and the rest, the 90%, there's no symptoms. I consider polio an incredibly well-adapted human pathogen. We'll get back to poliovirus later. Now, acute infections are public health problems. The big epidemics you hear about are typically acute virus infections. Despite the fact that everybody in the world's infected with herpes viruses of one sort or another, you really don't hear much about that. What you hear about are the acute infections like influenza, norovirus, and now Zika virus, which is infecting millions and millions of people. Last year, it was Ebola virus. These are public health problems in part because they happen quickly. By the time you're sick, the virus is already at its peak, and you've been shedding it and infecting people. And so just looking at people for illness isn't good enough to stop an infection. You have to have other ways to sort that out. And as I've said before, someday we're going to have ways of scanning ourselves by a scanner of some kind built into our mirror, I think. And every day when you get up, you will be told if you have any virus infections you need to worry about. And that information will be automatically transmitted to your physician, and you go pick up a prescription on the way to work. But we don't have that yet. I do think that will happen. We don't have it yet, and so acute infections are hard. And we will talk more about this issue when we talk about antiviral drugs. It's really hard to make antiviral drugs for acute infections, because by the time you're sick and then you go to your physician, you get a diagnosis. If you're lucky, you get your prescription, it's too late. All right, we have a question here, which is, which of the following do acute infections and incubation periods have in common? The virus is not replicating, no symptoms are visible, immune defenses are engaged, the immune system does not respond, or all of the above. All right, most of you got C, immune defenses are engaged, and that is certainly true during the incubation period. Of course, the virus is replicating, your immune defenses are responding. None of you picked A, because that's not right, that's good. Now, some of you said no symptoms are visible. And the question is, what do acute infections and incubation periods have in common? Now, an acute infection, yeah, the incubation period is defined not having a symptom, but as soon as you get to the acute phase, you've got symptoms, so that's not right. And of course, not responding isn't right either, because you are responding during the incubation period and during the acute infection itself. All right, so let's go over 5 viruses that cause acute infections. I took one out. You may have it on your notes if you downloaded it early today, because that was too many. I picked these based on different routes of transmission and different routes of spreading within the host. So we have influenza virus. Is a virus that's spread by respiratory secretions. It just causes a respiratory disease. We have polio, fecal-oral spread. Measles is spread by respiratory secretions, but it has a very different pathogenesis than flu. It has a viremic stage. Norovirus, fecal-oral spread, but a gastrointestinal disease, unlike polio. And finally, West Nile, a mosquito-transmitted infection. So this is relevant to Zika, same family, in fact. So let's start with influenza virus. There are 3 types of influenza, A, B, and C, and we only worry about A and C because C mostly causes either very mild or asymptomatic disease. And only influenza A viruses causes what we call pandemics. A pandemic is when you get an outbreak somewhere and it spreads throughout the world. Every country in the world gets infected, more or less. That's a pandemic. An epidemic would be an outbreak in the US or in a state of the US, for example. So only the A viruses cause pandemics. We're going to look at why later on when we talk about virus evolution and emergence. A hallmark of these viruses is antigenic variation. It's a way these viruses get around the adaptive response. They vary from year to year. Remember last time I told you that you can have a 1 amino acid change in the viral hemagglutinin. That's enough to make antibodies that you've made not very good at neutralizing it. And we'll talk more about that later on. You should remember influenza virus particles because we've looked at the icon before. On the upper right, we have the membrane-bound particle with 8 single-stranded RNA segments. And it's a negative-stranded RNA, of course. And on the upper left is a lovely 3-dimensional reconstruction of the particle showing the glycoproteins on the surface in blue and red, one of which, of course, is the hemagglutinin. And then the RNA all coiled up outside. And, of course, that's a negative-strand RNA. It's bound to proteins, so it's a ribonucleoprotein. So the virus is transmitted between hosts by respiratory secretions. It enters the upper respiratory tract. It begins replicating in your mucosal cells, the mucosal surfaces of the upper respiratory tract, and then can spread down into your lungs. And influenza virus can go all the way to the bottom of your tract and cause pneumonia. So when you get into the alveoli and start replicating in them, and they start to fill with fluid, this is pneumonia. That can be caused by influenza virus. And if we look at a cross-section of the respiratory mucosa, remember, we have I cut it off here, sorry. We have ciliated mucosal epithelial cells. There's a basement membrane. The virus replicates in these cells, causes inflammation, and spreads throughout the tract, but it doesn't get into your bloodstream. So it does not become systemic. It does not cause a viremia. It just spreads laterally within these cells. And as I said, it can go anywhere from the upper tract all the way down into the lower tract. Typically, when you get flu, you feel really crappy, right? And you get tired. You may have a chest burning pain. That's because the virus is really good at replicating in the trachea. And so it destroys cells in there, and you typically have a substernal burning pain associated with infection. So it's transmitted by droplets that you make when you cough or sneeze or just talk, and they're full of influenza virus. And these, remember we talked before, droplets, depending on their size, can drop right to the floor in front of you, or they can spread. The tiniest droplets remain suspended and can go long distances from 5 to 160 feet. So respiratory transmission is a main way that this virus goes from person to person, but you can also get it by direct contact with an infected individual. If you are shedding virus in your mucosal secretions, mucus, saliva, and so forth, and you contact someone directly, you can be infected. Or a contaminated surface. You can touch your mouth, nose, eyes. If you're infected, then you touch a doorknob and someone comes by shortly afterwards, they can get infected. A study was done in Switzerland a number of years ago which showed that you can put virus on Swiss banknotes where else would they do this experiment, right? Lots of Swiss banknotes changing hands apparently in Switzerland. And they pipetted flu virus onto the notes, and if they mixed it with mucus, which is how it would come from you to the banknote, imagine the teller, right, standing there counting your money out and picking their nose periodically, which people do. They're going to contaminate the money with flu, and that can last up to 7 days on the banknote if it's got mucus mixed with it. So that's a good way to transmit infection. So both aerosol and by contact. Now, a really important determinant of what kinds of flu strains you get infected with are the receptors for the virus. You may remember from a long time ago, the virus binds to sialic acids, which are at the ends of glycoproteins. And human strains bind to alpha-2,6 sialic acids. So if you look in the pharynx and the larynx and the trachea, you see lots of alpha-2,6 sialic acids present there, also in the paranasal sinuses. And human strains bind these, and that's no problem. They replicate very well up and down the respiratory tract. You can see lower in the tract, you have a mixture of alpha-2,6 and alpha-2,3. Now, avian influenza virus strains, these are viruses that just infect birds. So the source of all flu viruses in the world, whenever a new one comes to infect us, is birds, particularly aquatic birds. And in those animals, the receptor is alpha-2,3 sialic acid. So to go from a bird to human requires an expansion of receptor range. And if you notice here, alpha-2,3s are typically present lower down in the tract. It's always been thought that in order to get infected with an avian influenza virus, you have to inhale deeply and the virus goes all the way down into your tract. But it turns out that around your eye, there's lots of alpha-2,3 linked sialic acids. And so in theory, you could be infected with the virus in your eye, and then the virus could drain the lacrimal duct into your upper tract, and then encounter alpha-2,3 linked sialic acids there, and then they could go down into your lower tract. So this has actually been borne out in studies of people who work on chicken farms. They can often get eye infections that spread to different parts of their tract. Just an example of how receptor utilization can control where a virus infects. So a typical bout of uncomplicated flu, 1 to 5 day incubation period. All of a sudden, you get sick, and you can tell exactly when you get sick if you get flu. You get a headache, chills, dry cough, high fever, muscle aches and pains, etc. And the fever peaks within a day, and then it starts to go down, but the infection continues, as you will see. And this differs in children and the elderly, which typically have more severe disease. You get respiratory signs amplifying. So very early on, you have a high fever, but then as that goes away, you think, I've beaten this, and then you start to cough and hack and sneeze and spit mucus all over the place, and that can go on for quite a while, 1 to 2 weeks. The virus is replicating throughout your tract, again, depending on the receptors for the particular strain. All the human strains are going to bind to the alpha-2,6, and you're not likely to be infected with an avian virus unless you're working in a chicken farm or something of that sort. How do we diagnose it? Well, turns out if you go to your physician and you feel lousy in the middle of winter and you have fever, cough, or sore throat, that is called an influenza-like illness, or ILI, and they will diagnose you with flu, even though it could be caused by any number of other viruses. And if you want, they will give you an antiviral, even though you may not have flu. So this is the state of our diagnostics for influenza. Fever, cough, or sore throat, and no other known illness. We have rapid lab tests which can be done in the physician's office. They can take a little bit of mucus or do a nasal wash and put it on a dipstick-based test, sort of like the pregnancy tests where you get a strip illuminating if it's positive. And these are really crappy, about 50% accuracy. So if you want to make a contribution and you're interested in engineering and science, make a better diagnostic test for flu, because if they say you're not infected, they can be wrong half the time. You can, of course, diagnose very readily by PCR or growing the virus or looking for antibodies, but you can't do that rapidly, and most physician offices can't do it. So we have a problem in diagnosing, so they just say, you have flu, take this antiviral. Now, flu is one of those viruses where if you pick the right strain, you can actually do infections of people and then see what happens. You can have people come in, register for a clinical trial, and you can infect them and then follow them from day to day. In fact, there's a company in London that does this, and you can contract them to do it for you. They have a little hospital where they will take 20 patients and put them each in their own room, infect them all with virus. They will pipette a little virus into your nose, and then they will follow you. They measure your temperature every day. They come in and see how you feel, how much mucus you're producing. They weigh the tissues that you're blowing your nose into, and so it's grams of mucus per day. And they can measure virus production in your nose. They can measure antibodies in your blood and so forth. So these are curves from some of these studies where we've inoculated volunteers. Here in the top left, we're just looking at virus titer in the nasal wash over time. And you can see by 1 day, you're already getting quite a bit of virus produced. It peaks at 2 days, very rapid infection. And then in the open squares, these are the symptom scores. So they have a rubric for asking, how you feel, how much you're coughing, and so forth. And that is shown in the white boxes. And you can see that peaks slightly after the virus peaks, and then goes on feeling bad goes on for a long period of time. The bottom right is a little more detailed. Again, we're looking at volunteers inoculated with virus. We're looking at either virus titer or antibody titer on the y-axis versus days after infection. Again, you can see peak virus titer within 2 days after inoculation, then it goes down very quickly. Interferon, the dashed lines, you can see that goes up in parallel. So very early on, the interferon system is kicking in. This probably plays a big role in resolving infection. But then you see your adaptive response, nasal wash, neutralizing antibody, and serum neutralizing antibody. These are these dotted curves. You can see they come up way later. Look, they're starting to rise at day 7 or so when the virus is already gone. So they're not playing any role, these antibodies. Of course, the next time you get infected, they're going to play a role as part of the memory response. Now, flu is a seasonal disease. In temperate climates, which is what we are in, we have a summer and a winter. It occurs in the winter months. And this is a graph showing the number of cases of influenza virus. This is confirmed by viral isolation by CDC and associated laboratories, WHO laboratories. And you can see a nice peak in the winter months every year. The summer months, no flu or very little flu. Peak, peak, peak. This is from 2004 to 2007. And here we've broken down the viruses into type A or type B. So you can see you have a mixture of types every year. Now, this year has been a relatively mild flu season as flu seasons go. We had very little in terms of virus isolation. Again, the color codes on the bars represent A or B of different sorts. But you can see, so this is 2015, week 40. So that's 10 weeks from the end of the year. So 4-8, that's December, November, October roughly, right? And we should already have some good flu activity by then. We didn't have anything until really the first weeks of 2016. Here's 2016, week 2. Very little, 500 positive specimens. And then only very recently there was a peak of activity of influenza. And I talked to a friend of mine who's a clinician up at the medical center, and he said the emergency rooms throughout the city were full of people coming in with influenza-like illness. And we don't know why there was a sudden peak. My theory is that they're mostly old people who got immunized in August and their vaccine is already wearing off. But I don't know if that's true or not. Anyway, this was a very mild year. There's a great site run by the CDC. Flu is a reportable disease, and CDC has many laboratories throughout the country. In fact, WHO has global laboratories who take many people who report to hospitals and clinics with flu-like illness, and they isolate virus with them because they'd like to know how the virus is changing so they can predict where it's going in the next year. So you get a lot of data from this, and it's all at cdc.gov/flu. Here's some more of it. This is a very interesting curve of pneumonia and influenza mortality. So if you die from flu, typically they don't write flu on your death certificate, right? The cause of death is typically pneumonia or heart attack even. So they combine the two to get this idea of what flu is doing in terms of mortality. So what we have is the week of the year. These are different years, 2012, 13, 14, 15, and 16. And you can see the epidemic threshold. And again, the infection is seasonal, so it's peaking in the winter months. And the red bar is the number of cases of flu and pneumonia mortality. So these are deaths due to this in all age groups. And you can see there's an excess. This is called an excess number of cases over what would be predicted. The two black lines are a statistical prediction of the maximum and minimum number of deaths you would see every year based on the number of infections. And in some years, you exceed that substantially. In 2015, we did not. And so far, this is 2016 right here. I don't know what the end result will be. My suspicion is there'll be far fewer flu deaths because there's not been much infection at all. So that's another interesting statistic. Now, in 2015, this is the graph of laboratory confirmed flu hospitalizations. So this is throughout the US. You can also go to the New York City, the health department website. They give you the same information for New York City. So you can see here, this is different age groups, but the biggest curve is 55 years and up. So these are the people that really get sick with flu. They go to the hospital, they're admitted. And here you can see, what is this on the right, left here? This is a number per 100,000. And you can see it goes up to 280 per 100,000. And then the other age groups are down here. You can see the very young are also most susceptible. And then everybody in between, not very much. That was a year ago. And this is the curve for this year. You can see it's much lower. We're peaked so far at about 40 cases per 100,000, so substantially lower than last year. So again, not a lot of activity, which is good, of course. And we don't know why this is. It could be the vaccine is spot on this year. It might be the virus isn't circulating. Who knows? We don't really understand this. So typically every season there are 35 to 50 million cases of flu a year, and anywhere between 3,000 and 49,000 deaths. That's an estimate based on our experience of the past 30 years or so. So this is why we have a flu vaccine. It really makes people sick and kills a lot of people. Besides the respiratory infection, the initial infection caused by the virus, there are complications which include pneumonia. That doesn't happen in every case, but in particular, elderly people are prone to get pneumonia. You can get secondary bacterial pneumonia. After the virus is gone and you think you're okay, all of a sudden, bacteria start to cause a problem. Generalized muscle pain, sometimes cardiac involvement, and Reye's syndrome. Now, there are some interventions, as I mentioned briefly. There are non-pharmacological things you can go to the drugstore and waste your money on. If they make you feel better, that's great. They're probably not much better or worse than the antiviral drugs. And we have a couple of them, Tamiflu, which is taken orally. It's a pill, Influenza, which is inhaled. And we'll talk about these later. These are inhibitors of one of the viral enzymes. And then rimantadine is an inhibitor of virus entry. And it inhibits the proton pump in the particle that's important for virus entry. We'll talk about that later. And of course, there are flu vaccines, but as we will discuss later, they are problematic for a number of reasons. But you should get them anyway, because if you don't, you have a higher likelihood of being infected. I'm just curious, no judgment here. I'm wondering, how many of you got a flu vaccine this year? I did. That's not bad. You know, you're lucky you're young and you're not going to die, likely, unless you have some immune defect. But if you're 60 and you're not getting the flu vaccine, well, I'll be dead by then, but my words will haunt you, okay? All right, number 2. Which of the following is characteristic of uncomplicated flu? Transmission may occur via respiratory droplets, incubation period 1 to 5 days, fever peaking in 1 day, coughing and weakness can last for 2 weeks. Or all of the above. All of the above is correct. Most of you got that. Okay, the next acute infection is caused by poliovirus. Polio is a RNA-containing virus plus stranded RNA, which is encased in a lovely icosahedral shell made of 4 different proteins. There's an EM of the virus. It's about 30 nanometers in diameter, kind of small. And there is a reconstruction from the structural data on the bottom. This virus is acquired when you ingest fecally contaminated material, which we do all the time because, as a famous virologist once said, the world is covered with a thin layer of shit. And they're right, because we don't do a very good job at taking care of our waste. And we don't wash our hands. We go to the bathroom, we don't wash our hands. We have fecal material on our hands. We contaminate the door, everything, and you infect other people. So you ingest virus, it goes down into your intestinal tract, it replicates in the mucosal cells, it passes beneath the basement membrane. So this virus does establish a viremia, goes through the lymph system into the blood, and you get a viremia. And from there, it can go to various tissues. But again, only in 1% of people does it get into the CNS. We think it gets in by infecting muscle. The virus is brought to the muscle by the blood. It then enters the motor end plate, the end of the nerve within the muscle. It travels up the nerve by retrograde transport and then replicates in the motor neuron in the spinal cord. And if it destroys enough motor neurons within the cord, then the limb innervated by that area of the cord gets paralyzed. But that's only 1% of the time, and we are not sure why that's the case. I think those 1% have a SNP in some gene controlling innate responses, but nobody has looked at that. Now, the key here is that the virus replicating in your mucosal surface of the gut is then shed into the lumen and into your feces, and it passes on to other people. This virus causes no pathology in the gut. You have no gut pain, you have no diarrhea, nothing, no gastroenteritis at all associated with polio infection. The only thing is the accidental invasion of the CNS. So I view this as accidental because if you think about it, the virus has no way to get back out of the CNS. So this is a dead end from an evolutionary viewpoint. Now, if this were an animal, and animals may eat each other, you can imagine that that could spread the virus from one animal to another. My understanding is that animals like to eat each other's brains. They're very tasty. So, in particular, animals with strong jaws. So that would be a way of getting virus from one to another, but we don't do that. So I think it's an accident. The main route of transmission is by the gastrointestinal tract, and it doesn't cause any disease. You don't even know it. If it weren't for the paralysis, we wouldn't even be immunizing against polio. We probably wouldn't know about it, and it would silently spread very effectively from person to person. So that's why I think this is a beautifully adapted pathogen. So in our transgenic mouse model for polio, which I talked to you a couple of days ago about, this is a section of spinal cord taken from one of our mice that was paralyzed after injection of virus. And it is stained green. Wherever there's a green dot, that's where the virus is replicating. So it's a section of the cord. And these green dots are poliovirus RNA. And those are neurons. The virus is replicating exclusively in neurons. And again, it destroys them, and you get paralysis. And all these other dark cells are probably macrophages of some sort coming in trying to clear the infection. So again, you destroy enough neurons in an area innervating a particular muscle, and that gives you the paralysis, which is so typical. This is a very neat diagram from a 1950s textbook of medicine. Now, if you go to medical school, you won't have any polio in your textbooks of medicine anymore because it's been eradicated from the US. And so now and then there's an imported case, and no one knows what it is because no one is trained anymore to recognize polio. It is drawn by hand also in the old days. And what it is a combination of observations of many different patients where we're looking at a variety of measurements on the left with time after exposure to virus. So for example, on the top is temperature. You can see there's about a 7-day incubation period. You have no symptoms. That's the definition of the incubation period. And then you have a fever right there, that peak in temperature. You also get a headache, sore throat, nausea sometimes, but not all the time. And then in most people, 99% of the people, they get well, and then nothing ever happens. However, at the same time, they are shedding virus in their feces and in their throat secretions. You can detect virus in their blood, so there's a transient viremia. 1% of these infections, about 5 days later, 12 days total incubation, you get another spike of temperature, much higher temperature at that point. You get headache, nausea, stiffness, pain, and paralysis. And there, the virus has invaded the CNS. This bottom panel is virus in the CNS. So you can see in the preliminary illness, the non-specific illness, there's no virus in the CNS. Only when the virus gets in the CNS do you have the paralysis. So that's at about 14 days post-infection. So here's a nice illustration of why it takes time for the virus to get from your gut into the CNS. So if you looked at the incubation period as just the time to paralysis, it's 14 days because the virus has to travel there from the gut. So we are the only known reservoir of infection in nature. The virus doesn't infect anyone else. It's spread by fecal-oral transmission. There's a seasonality in warm months and temperate climates. You don't really know why this is, but when there were outbreaks of polio in the US, kids would typically get it in the summer months. There is a complication called post-polio syndrome, which happens about 30 to 40 years after having your initial bout of polio. So if you get polio and you have leg paralysis, that can be restored with therapy. You can be walking again. Of course, the virus can target neurons that innervate your heart and lungs, in which case you can die unless you're caught really early on. But if it's just paralyzing your leg muscles or arm muscles, you can have function restored. But then in about 25 to 40% of those people, 30 to 40 years later, they start to get paralyzed again. There's no virus present. It just seems that you've lost many neurons as a consequence of the first infection. And as you're aging, you're slowly losing neurons. And if you haven't had polio, this is not an issue. But if you've lost a lot of neurons, then you don't have enough of a backup, I would say, and you have this post-polio. So we still have people today with post-polio because up until the 50s, the end of the 50s, we had polio in this country. Of course, eventually that will stop, but these are still individuals that have to be cared for. So in the US, we used to have 20 to 30,000 cases of paralytic disease every year. We introduced an inactivated vaccine in the 50s and an infectious vaccine in 1960. And now the last case of polio in the US was 1979. It's now been eradicated from the US and, in fact, much of the world. And we'll talk about that in more detail when we talk about vaccines. But again, this vaccination, the development of vaccines, was solely because the virus causes paralysis. If it were not for that, no one would care because it's an asymptomatic or mild illness. Okay, so that's an example of a virus that you acquire by fecal-oral contamination. Replicates in your gut. It spreads to other sites within you by the blood and has a very rare complication associated with it. And one thing I want to point out, which is interesting, back in the 30s and 40s, when polio infections were at their peaks, here in New York City, a common practice was to quarantine kids who had polio. The police would go from door to door and look for kids with paralysis because the parents would hide them. They didn't want anyone to find them because if the police found them, they would take them and bring them to a hospital and quarantine them for a few weeks. They'd take them away from their family. And this was a zero-use practice. It had no effect whatsoever. Can you tell me why it was useless? How many people get polio out of the total infected?
Just because you have polio doesn't mean you get paralyzed.
That's right. What's the number? Do you remember? It's 1%. So 99% of kids were infected, walking around shedding virus. Those are the ones spreading infection, not the kids who were home paralyzed. It was absurd, but we didn't know that at the beginning. And finally, we figured it out, and they stopped doing that. But quarantine for polio in the early days was useless. Okay, measles virus. We have mentioned briefly. It's a member of the paramyxovirus family. It's an envelope virus with a negative strand RNA, but unlike flu, it's a negative strand in one piece, quite long, and this virus has glycoproteins in its envelope as well. And this, I used this photo very early on to show you a helical nucleocapsid within an envelope, because here on the left is an electron micrograph. You can see the lovely repeating nature of the single helical nucleocapsid. So this is one of the most contagious human viruses we know of. Anybody see this movie, Contagion? No? Yeah, a few of you. So there's one point where, I forgot her name, she was playing someone from the CDC. She's writing on the blackboard R-naughts of different viruses. And that's what this number is here, R-naught. That's the number of people you can infect when you're infected with a particular virus. And so for measles, it's 15. So on average, this is on average, based on studies that have been made, any one person infected with measles can infect 15 different people. And you need a very large population to maintain epidemics of this virus infection. So there's one serotype, and if you get infected with it, you have lifelong immunity. And in fact, those were the early experiments on the Faroe Islands that I talked about a few lectures ago that proved that memory exists. It's transmitted by respiratory particles full of virus, much like influenza virus. But look at this. You are infected for you are infectious a couple of days before the rash appears. And a kid typically infected with measles may not have any symptoms before the rash, maybe a mild fever, but nothing that would prevent your parents in the old days anyway from sending you to school. Nowadays, if your temperature goes up 1 degree, they send you home so that you won't transmit infections. But in the old days, that wasn't a big deal. And so kids used to spread measles very effectively. Of course, this was pre-vaccine era. Now we immunize and we've gotten rid of it. This is an interesting infection where everybody who gets infection shows disease. There are very few asymptomatic infections. How the virus spreads within you, you inhale respiratory aerosols or put virus in your mouth, similar to flu with contaminated fingers. The virus does not replicate in the mucosal epithelium. It actually replicates in immune cells that are patrolling your mucosal surfaces, like monocytes. And that's shown in the upper right here. So we have respiratory epithelium. I've expanded that on the upper right. And the virus particles you can see in blue, but the orange cell, it's like a dendritic cell, right? They get infected, and then they bring the virus below the respiratory epithelium. So the virus doesn't initially replicate in your respiratory mucosa. It's brought in by dendritic cells. The dendritic cells, they go, of course, to the lymph node to present these antigens to T cells and B cells, and that ends up releasing virus, and they infect. The virus replicates in lymphocytes, so that's a perfect place for the virus to be brought. It replicates there. You get a primary viremia. The virus gets in the blood and then goes to a variety of other organs, which we'll talk about in a moment. One of those, of course, is the skin, giving you the characteristic rash of measles. Now, how do you spread it? So the virus is multiplying at various places. There's a secondary viremia, of course. Lymphoid cells that are infected traffic to the respiratory epithelium. So we're back in the lung. So here was the lung cell surface on entry, and now we're back at it for exit. And the immune cells in which the virus is replicating bring the virus to the basolateral surface, and that's where the receptor, or one of the receptors for measles virus is, Nectin-4. The virus gets into these cells, replicates, and is released at the apical surface. So this receptor isn't on the apical surface of cells. That's why the virus doesn't get in that way. It comes in through a dendritic cell. But when it is released, the virus is released at the basolateral surface and can get in from the bottom. It's really an amazing strategy, and that allows this blood-borne infection to be spread by respiratory aerosols, because these virus particles that are released at the apical surface, they, of course, are then expelled by you when you cough or sneeze, and that's how you spread it to other people. So the cardinal sign of measles infection, of course, is the rash, which is shown on this child here at the right. You have a high fever, you have respiratory system, runny nose and cough, conjunctivitis. The rash is all over the body. And then in the mouth of these children who are infected, there is a very specific sign called a Koplik spot. And these are whitish areas on the buccal mucosa. So here's the tongue over here on the lower left, teeth, and these are the white spots here. If you were to take a little bit of one of these white spots and look under a microscope, you would see syncytia, big giant cells full of many nuclei, which we talked about way at the beginning. When measles virus infects a cell, it produces its glycoprotein on the surface, and these cause neighboring cells to fuse. So this is diagnostic for measles virus infection. And there are substantial complications with measles virus infection. Encephalitis, 1 in 1,000 cases. Virus gets into the CNS and destroys cells there. Bronchitis and pneumonia, ear infections, and fatality. In our country, in the US, before the vaccine, the fatality was 1 to 2 per 1,000 cases. In countries with poor nutrition, 28%. I mentioned this the other day. Measles becomes very lethal in areas where the kids are not properly nourished. You have a long-term sequelae called subacute sclerosing panencephalitis. We will talk about this next time, because this is a form of a persistent infection. And remember, this virus is one of those that causes immunosuppression. It infects cells of the immune system, including the antigen-presenting cells, and immunosuppresses you so that you get infected with other things besides measles virus. And in third world children, this is a main cause of death. It's not measles itself, but it's getting infected with everything else that they have in these areas. Now, before the vaccine, we used to have 3 to 4 million cases of measles in the US, 400 to 500 deaths. I had measles as a kid. We didn't have a vaccine when I was growing up. 400 to 500 deaths, 48,000 hospitalizations, and then every year 1,000 kids would have chronic disability from having encephalitis, virus replicating in their brains. We stopped endemic transmission in the year 2000 by the vaccine. The vaccine is given as part of a trivalent preparation, measles, mumps, rubella vaccine, MMR. Very famous vaccine because in 1988, a British gastroenterologist by the name of Andrew Wakefield published a paper saying that measles vaccine caused kids to get autism. All right? Now, this paper was subsequently retracted because it's mostly fabricated, and he had a number of conflicts with insurance companies and so forth. It's not been reproduced anywhere. There's no connection between this vaccine and autism, but as a consequence, many people decided they would not vaccinate their kids. So they took the same course of action as the governors of New York and New Jersey. They decided they were doctors and didn't listen to the advice of their physicians. And we had outbreaks in the UK and Ireland, many other places. And of course, in the US, we also have people who don't want to immunize their kids with MMR. And so we have outbreaks as well in the US. Now, Wakefield lost his license to practice medicine in the UK. He ended up in Texas, of all places. They welcomed him. I don't know what he's doing there, but one of the things he's done is he's made a movie, apparently, called Vaxxed, which says that this vaccine, he still contests that this vaccine is bad. And they were going to show this in the Tribeca Film Festival in a couple of weeks. You may have heard this. Unbelievable. Now, Robert De Niro, the very good actor, I think he's responsible in part for picking the films. He said, well, they have an autistic kid, and he said both sides of this should be aired, and that's why he wanted to show the movie. Well, I got news for him. There are no both sides. There's only one side, and that the vaccine is safe. It does not cause autism. So for them to show this is just unconscionable. Fortunately, over the weekend, somebody convinced him to pull it. So, you know, Wakefield can make whatever movie he wants, but it cannot be showcased because he's wrong. And there's nothing wrong with the MMR vaccine. It's absolutely safe. It prevents many, many millions of infections a year, and it saves lives. Here are the numbers. Vaccine licensed in the US in the 60s. We used to have hundreds of thousands of cases a year, hundreds of deaths a year. It eliminated all of that, almost in the US, except for the people who don't immunize their kids. We continue to have outbreaks. The most famous one started in Disneyland in California. You know, a bunch of parents who didn't immunize their kids, they went to Disneyland and they were infected, but they didn't have a rash yet, so they spread the infection. Ended up with 178 cases in 17 states. It spread all over the country. And we've had cases every year in people who don't immunize their kids. These are the numbers. And that's the spike in that year, 2014 to 2015. All because of parents who don't immunize their kids. It started in California. It spread to all these other states in the US. Totally inexcusable. Now, globally, we still have a lot of measles. These are the reported measles cases according to WHO from August 2015 to January 2015, only 6 months. You can see some countries still have over 1,000 cases. 7 countries, 4%, have a lot of measles. We have some in the US because of people who don't immunize. But much of this is because we don't get to these kids and immunize them properly. And this is a big goal of WHO. In 2014, there were 115,000 deaths globally from measles, and between 2000 and 2014, 17 million deaths were prevented. And this is one of the best buys in public health, the measles vaccine. In addition, because measles is immunosuppressive, it turns out that kids who get it don't get other infections as well. All kinds of bacterial and parasitic infections, you spare them. This has been studied, and it's quite clear that this is a terrific vaccine. There are very few side effects. There are no downside to giving this to your kids. So I do not understand why parents would listen to actors And remember, Jenny McCarthy was another big actress who spoke out against MMR vaccine. I don't get this. It doesn't make any sense. If you have any hesitations, please throw them away. There's nothing wrong with this vaccine. Okay, so we have a question related to this. Which of the following is a good reason to get measles vaccine? 1 in 1,000 chance of encephalitis, 1 to 2 in 1,000 chance of death. Each infected person spreads measles to 15 others. Immunosuppression can lead to secondary infections. 98, that's about the highest we've got so far. All of the above, that's right. Let's talk about gastroenteritis. Did you know that in any 24-hour period, the amount of water that people pass because they have diarrhea equals the amount of water passing over Victoria Falls in 1 minute? 65 million liters per minute. That's a lot of diarrhea. And that's not good if you're somewhere where the fluid can't be replaced. And so a lot of kids have died of this in countries that can't do fluid replacement. I want to talk about one virus that causes gastroenteritis, and that is norovirus. And this is a small plus-stranded RNA containing virus, which is a member of the Calliciviridae. We haven't talked about this family really because we use polio as a model for plus-strand viruses. This virus is responsible for half of all the foodborne outbreaks of gastroenteritis, 23 million cases a year in the US and rising, as our food preparations become not only globalized but institutionalized as well. We don't cook as much as we used to at home, and we end up getting food poisoning. So here's a graph which is a bit old, but it shows you norovirus, 49% of known causes of foodborne illness, and the CDC and USDA, the US Department of Agriculture, they track this very carefully because they want to try and encourage good food preparation practices. See, bacteria, 40% of the outbreaks. Before norovirus was discovered, which wasn't that long ago, we had no idea that viruses were such a big cause of foodborne outbreaks of illness. This virus is spread by fecal-oral contamination. You ingest fecally contaminated material. There are some other ways of spread as well. It passes through the stomach, replicates in your intestine. You get a blunting of the villi. Here's on the left, lower left, these are wild-type villi in the intestine, and on the right is someone who has been infected with norovirus. You can see they're blunted. Again, this is a virus you can do volunteer experiments with. In fact, they often use medical students to do this. They infect them on Friday, and they get sick over the weekend. They collect specimens, and then Monday they can go back to class because they're better by then. They get $300 for that. And the thing is, up until very recently, you could not grow this virus in culture. So they gave them stool extracts. Filtered, though, of course, but they flavor it so it doesn't taste so badly. It's got to be worth $300, right? We don't know why you vomit and diarrhea, so you have both vomiting and diarrhea. I call this a two-bucket disease, right? You need 2 buckets to collect. We don't know why the virus does this. Here's a very cool observation made, I think, last year. For years, nobody could grow norovirus in culture. Nobody could figure out what cells to use, what was going on. Now, how are they doing this? They would take diarrhea from someone with a neuroinfection, and they would filter it to get rid of the bacteria, and then put it on cells, and nothing would ever grow. Well, guess what? One day, someone forgot to filter it, and they threw it on cells, and the virus grew. It turns out the virus needs bacteria in order to grow. Here's the experiment. This is virus growth with days in cell culture. They grow the virus in B cells. Apparently, in the subepithelial space, there are B cells in which the virus grows. If you filter the stool, it doesn't grow. If you do not filter it, it grows very well. And we think that some component of the bacterial wall is actually stabilizing and binding to the norovirus and allowing it to be transcytosed across the intestine and to get into the B cells. So now we can grow these viruses in culture. These viruses infect everybody year-round with some peaks in cold weather. They typically occur in semi-closed environments, which we're making more of course, particularly cruise ships, but also all kinds of schools. Anything that favors person-to-person spread. There are outbreaks on campuses all the time, particularly associated with food preparation. There's a short incubation period, sudden onset of vomiting and diarrhea. It's a rather short illness, so over the weekend type of illness, although if you have immune defects, you will be sick for a longer period of time. And one of the problems here is that there's a 30% asymptomatic rate, which means if you are a food preparer, you can be shedding virus, and if you don't clean your hands well, you will contaminate food, and then you will cause a big outbreak, whether it's a restaurant, a cruise ship, or a hotel. And that's why there are signs in restrooms. Employees must wash their hands. The next time you go in and you see that, you have norovirus to thank for that, because that really raised awareness of what was going on here. This is an interesting graphic from the CDC, which makes a point that I wanted to tell you. You don't need many virus particles to get infected. They've done some studies with volunteers. You can be infected with 18 virus particles. So that's one of the most infectious viruses we know of. I'm not talking about how many people you can infect, but just the number of particles you need to infect a new person. So a little bit about the pathogenesis. The shedding peaks a couple of days after the onset, but it can persist in certain individuals for over a month. Even when they're better, they can continue to shed. So they go back to work and they start another outbreak. Besides fecal-oral transmission, it's also been shown to be transmitted by the aerosols produced by vomiting. So when you vomit, you make an aerosol, even if you're trying to contain it. And those aerosols have particles in them. Contact with contaminated material, food, water, or environmental contamination. So if you are putting fecal material into the water system, untreated, unprocessed sewage, you can transmit infections that way. Foods can be contaminated. For example, oysters, which are filter feeders, will take the virus up from the environment if in fact you're putting it in there. It's often present on fruits or by food handlers, as I've already mentioned. And for reasons we don't understand, you get infected, you recover, and then next year you can get infected again. Even though there are multiple genotypes, but there's typically a predominant genotype which you can get infected over and over with, and we really don't understand that. The best treatment is to prevent dehydration, take fluids. Humans seem to be the only animal that are infected with this, although there's some evidence for an animal reservoir. We do have a vaccine in early development, so I suspect at some point we will give vaccines to people who work, for example, in food preparation, on cruise ships, in hotels and so forth, just to interrupt that mode of spread. Everybody doesn't need to get this vaccine, but if you're at a point of risk, you should. And overall, this is not a serious infection, except if you're immunocompromised. Most people, you will recover and move on. What kinds of settings are associated with outbreaks? Here on the left, settings, you can see restaurants and catered meals, 40%, retirement centers, nursing homes, hospitals, 25%, schools, daycare centers. Vacation setting, including cruise ships, only 10%, but they get a lot of press, of course, and that's why you hear about it. Mechanisms of transmission, person to person, 88%. So most of the infection is transmitted by fecal contamination, one person to another. The foodborne and waterborne are really a minority of that. Here's an article from a newspaper where an entire flight was infected with norovirus. Someone threw up on the plane because they had norovirus infection. They aerosolized the virus, and everybody else in the plane got infected, including the attendants who went on to another flight, and they infected everyone else on the other flight as well. So quite a infectious pathogen here. Now, cruise ships also have outbreaks of norovirus infection. These are just some different cruises that have been, and here are the dates they went out, and they had outbreaks of gastroenteritis. Some of them were norovirus, but there are clearly some other agents out there that we don't know about. And for a cruise ship, this is a big deal because if there's an outbreak, it has to come back. Everyone has to get off. They have to decontaminate the whole ship, and this is very expensive, and they don't like that at all. So they try to go through really extensive means to try and prevent infections. Why are they associated with cruise ships? It's only because we track the illness particularly. As I showed you on that pie chart, there are not a lot of them, but the close living quarters, of course, increases group contact, and new passengers come in and out, and they can spread the virus as well. So personally, I would never go on a cruise because of this. I think you got to be crazy to risk getting this, but it really isn't that predominant. All right, so how do you prevent a norovirus infection? Wash your hands, rinse your fruits and vegetables, cook your shellfish, and if you're sick, don't prepare food, and please do not eat the mints in the restaurants. Because people go to the bathroom, they don't wash their hands, and then they grab a mint, and people have done studies. These are full of fecal coliforms. It's fecal contaminated. Even if they have nice wrappers on them, don't take it because the wrappers are contaminated. It's a nice way to get sick. Norovirus transmission could be curbed by covering your mouth when you sneeze, washing your hands after using the bathroom, using condoms during sex, using mosquito-proof nets, or none of the above. You got the feces message, I guess. That's right. Wash your hands. Okay, the last virus is West Nile virus, which causes an illness to make you look like a pharaoh. Not really. Isolated in 1947 in Uganda, same place where Zika virus, same family of viruses, was not in the Western Hemisphere until 1999. Came to New York in August 99, and from there has spread throughout the Western Hemisphere. Now it's in North and South America, as well as in other countries where it was previously. Important thing here, it infects hundreds of birds and many different kinds of mosquitoes and other vertebrates. So there's a big reservoir for this virus besides humans. It is a flavivirus, enveloped plus stranded RNA containing virus, and we've talked about these a bit in the past. The transmission cycle in nature, the virus is brought among birds and other animals by mosquitoes, in particular Culex mosquitoes. And so out there, the birds are infected, the mosquito brings it from bird to bird, and when humans intrude upon this cycle, they become incidental hosts. So the virus does not spread from one human to another, but from birds to humans, and so forth. Horses may also be infected as well. So it's transmitted by culicine mosquitoes, incubation period, 3 to 14 days. 20 to 30% of people develop a flu-like illness called West Nile fever, relatively mild illness. 80% of people have no symptoms. So not a really serious, but when you talk about CNS involvement, it becomes more serious. 1 in 150 people develop neuroinvasive disease. The virus gets into the central nervous system, and you can have a whole host of problems associated with that, including headaches, even polio-like flaccid paralysis, 10% mortality, so it's a serious illness. And half of these people have long-term neurological sequelae. This is the reason we're making a vaccine for West Nile, because of the neurological sequelae, because these people have issues, long-term issues. And an interesting thing was this wasn't really appreciated until it had spread globally. So how does the virus infect you? So we have a bird-mosquito cycle. Mosquito bites a human. Here's the skin here, and the virus delivers the mosquito delivers the virus into the skin. It's picked up by dendritic cells, which is their job. However, the virus replicates in dendritic cells. The dendritic cells go to the lymph node, and as they're presenting the epitopes to the lymphocytes, the virus is replicating, and it finds more cells within the lymph node in which to replicate. It establishes a viremia, gets to other organs like the spleen where it replicates extensively. Again, that's the major infectious pattern. A whole variety of immune defenses are required to clear infection, including T and B cells, interferon and complement. But again, as I said, in a small percentage of people, the virus gets in to the brain, which is shown at the bottom of the slide, and infects neurons, and that causes the neurological sequelae. As I showed you, I think, 2 lectures ago, we think that sensing of the virus by toll-like receptor 3 causes production of TNF-alpha. That loosens up the blood-brain barrier and allows the virus to get in. So this is the part in the neuron, in the CNS, that we are trying to vaccinate against. In the CNS, once the CNS is infected, immune cells then come in from the periphery, including CD8 and CD4 positive T cells, and these are essential for clearance of the infection. Now in the US, since 1999, when the virus first came, you can see the number of cases a year, sometimes as many as 10,000 cases. The orange bars are the deaths. The dark purple bars are the neuroinvasive cases, and then the rest are the total. So some years, very little, and other years more than others. And we don't know what causes this. We do think there's some correlations with weather. When the weather is particularly dry, the birds leave particular areas because there's no water, and mosquitoes feed on humans instead. That's one of the theories that's been put forth. Just to give you an idea of where these infections are in the US, in 2012, there were 5,300 cases, pretty much scattered throughout the country, 2,700 neuroinvasive cases, and 243 deaths. The CDC also tracks West Nile infections. You can get a lot of information about that on their website. This is the incidence of neuroinvasive disease by age group from 1999 to 2014. And you can see that it's mostly elderly people who are susceptible to neuroinvasive disease, much less of a problem in young people. So there's something about them. Maybe their TLR3 response is aberrant. Maybe the barrier to the CNS is aberrant. We really don't know what the problem is. This is also a seasonal disease. You can see this is a summary of all the cases from 99 to 2014, grouped by month in the US. And you can see there's a peak in the summer months. And of course, this is because that's when there are mosquitoes in most of the country. So here in New York and New Jersey and Connecticut, you don't have any West Nile in the winter. And now that the mosquitoes are beginning to breed again, we'll see it would typically start to see cases of West Nile. Whether we'll have a lot of cases this year or not, we don't know. How do you prevent infection? For now, what you have to do, just like for Zika virus, is to avoid contact with mosquitoes. You have to get rid of standing water around your home so the mosquitoes can't breed in it. You have to be careful about how you're going outside when mosquitoes are biting. You have to cover yourself because we don't have any antivirals and we don't have any vaccines. As I had said, a vaccine is in development, and we'll probably have it out in the next 10 years or so. I'm not sure that everybody needs to get that, but if you're in an area where mosquitoes are feeding year-round, for example, might be a good idea to get that.